First of all, I want to thank you for inviting me to speak to you. I may be the only pediatric ID person in this conference hall, but there are more similarities than you might think. As a resident, I was always tickled by how vulnerable human babies and piglets were to rotavirus A, for example. And if you ever go to the state fair, you realize that holding piglets is a close second to holding human babies. So probably a lot more correspondences than one would think. But what I'm hoping to do is to show you more significant correspondence between my world in the hospital and yours as producers and veterinary caregivers. And I want to preface this. Forgive me, I understand that most of you are veterinarians, but there are many of you who are producers. I will try not to indulge in too much jargon, but please stop me if I do. So in this talk, I will attempt to do this in two ways. One is to describe the common world of antimicrobial use and antimicrobial resistance in the world that we inhabit together, both as human care providers and animal care providers, and also describe then in the skeletal form what I do as an antimicrobial steward in my hospital, and how such a hospital program might parallel what you do under very different circumstances. So I entitled my talk, Antimicrobial Resistance and Stewardship, What Farm and Hospital Can Share, both good and bad. Obviously, we share a lot of bugs, share a lot of resistance genes, but we may also share some solutions. I'd like to start with this because I think it outlines a metaphor that I think is injuring us when we consider the problem of resistance. Penicillin was a game changer. It was a wonder weapon of World War II. Some of you may not be aware of this, but Allied soldiers died of wound gangrene at a rate of, German soldiers died of wound gangrene at a rate 20 times greater than Allied soldiers because of the presence of penicillin. Not that the Germans didn't know about penicillin or have some access to it, but the Allied soldiers had access to quantities that were made available by the fact that there was a unique relationship, a remarkable coordination between pharmaceutical companies, research labs, and government agencies that allowed the production of pounds and pounds of this stuff. I understand that the original isolation of penicillium was very difficult. Thin film on a culture plate in Alexander Fleming's lab. By the end of World War II, they were making tons of the stuff in fermentation vats. But one of the problems with this is that, we were going to war on microbes. And some people consider the effort during this war as being second only to the Manhattan Project in terms of the scope of what was attempted to be done on a short-term basis. But the problem is it's a misleading metaphor. Microbes will never be eradicated, and it's a war we can't win. Microbes cannot be eradicated, and resistance will never be fully overcome. As you know, antimicrobial resistance far preceded us. There are samples of bacteria from permafrost 40,000 years ago that show many of the resistance genes we see today, and probably these resistance genes will be present long after we're absent as a species. So microbes and humans participate in a natural process occurring on a global scale. Some of it compared what we are undergoing in terms of antimicrobial resistance as being akin to what we're encountering with climate change. We're participants in natural processes which can be made worse by human actions. Our use of antibiotics accelerate the pace at which antibiotic resistance emerges and spreads in bacteria through both evolution and selective pressure. So no talk on ASP on antimicrobial stewardship is complete without the obligatory CDC slide. This is one national summary data from 2013, the latest that I have, which show that over 2 million people encountered illnesses due to resistant bacteria, of which there were 23,000 deaths. Since 2013, the problem has only gotten worse, I can assure you. There are new bad actors emerging. We have seen community-acquired MRSA and the emergence of ESBL, Enterobacteriaceae, in my world, such that now ordinary UTIs in children who have little to no antibiotic exposure are sometimes rather difficult to treat. Also, since 2013, we're appreciating the presence of carbapenem-resistant organisms. And for me, carbapenems are the last big gun. Once you blow through a carbapenem, you're dealing with some very interesting antibiotic choices indeed. So the problem with the CDC slides, it only shows the impact on the human of all these resistant bacteria. But I want to pull back and just put us in a greater, put things in perspective. And the fact is that the world's microbiome is gigantic. There are greater than 1 million species of bacteria in all habitats, of which maybe 10 to 20 are specialist human pathogens. Pathogens obviously like TB and Salmonella typhi, which are restricted to humans, and others which affect both humans and animals, and others, of course, many others might affect people who are immunocompromised. All of these bacteria engage in global crosstalk. This starts with intrinsic resistance genes that were obviously there as long as bacteria are making antimicrobials to control each other's growth. But understand that these genes have now been packaged in ever more efficient ways, allowing transfer in what are called mobile genetic elements, MGEs, such as transposons or plasmids, leading to horizontal gene transfer. It's kind of interesting that actually in 1944, the concept of DNA was first hinted at with transformation experiments by Avery, laying on raw, what they didn't know then was DNA, on the surface of pneumococcus. Basically, bacteria are very good at sharing genetic information, both within species and between species. And the real danger is they get really good at commingling and co-expressing both resistance and pathogenicity genes. The pathogenicity genes are the ones that allow certain clones of, for example, MRSA, methicillin-resistant Staph aureus, to spread worldwide within 1 to 2 years. I don't know what this says to you. This is a 20 million gallon manure lagoon, I'm told, the whitetail hog facility in Missouri. But I look at that and I think of it as a giant bioreactor, basically where many, many bacteria get together to share genetic elements. The sources of antimicrobial resistance in humans, as you know, often are the commensals, or the non-pathogenic bacteria that they carry within their nose, throat, gut. For example, 2 of the ones that I've been intimately involved in the last 20 years, one penicillin-resistant pneumococcus, the other community-acquired MRSA, are actually derived from transfer of, for example, the MEC-A cassette from coagulase-negative staph to Staph aureus, which allowed non-pathogenic skin bacteria to create a very, very pathogenic human organism. Soil is also another site of exposure, along with effluent in the water. I mean, it should be noted that bacteria they circulate and they circulate and they circulate. It's been estimated that there is 50 tons of bacterial biomass per person worldwide. And I think it's very interesting now, there actually is research to show that there's a wealth gradient, that in a place like India, the likelihood of infection with multiple drug resistance bacteria is directly related to socioeconomic status, which points to the role of cumulative environmental exposure. Food animals, as you know, can be a source of antimicrobial resistance. In humans, the presence of resistant organisms in the gut. This has been shown through shared genomes found in poultry and swine. The Salmonella typhimurium DT104 is a very good example of this. And then we jump in and make things worse. We give our animals antibiotics, we give our sick humans antibiotics, and sometimes we give our really not sick humans antibiotics. It's estimated that between maybe up to 50% of outpatient antibiotic use, it has no basis. Well, what about antimicrobial burden? This actually was shocking to me, knowing how stable antimicrobials are in the environment. Something like 175,000 tons of antimicrobials are produced yearly worldwide. Percentage devoted to food-producing animals varies by country. In the U.S., perhaps 80% food related. In less developed countries, around 50%. Give you an idea of where I stand in this big picture. Of the 20% devoted to human use in the U.S., only about 0.4% is used in a pediatric inpatient setting. And these antimicrobials don't go away. They circulate, they circulate, they circulate. They're stable in west effluent. And I'll show you this cartoon, which you've probably seen many times before, which show that we are actually in an inescapable web of ongoing antibiotic exposures. The organisms recirculate. The genes they carry, even should they break down, the genes they carry recirculate and are shared with other organisms. And the antimicrobials we use are remarkably stable in the environment, being not being broken down by waste processing methods. So you see here in this cartoon the fact that human antimicrobial resistance, organisms with resistant organisms are passed from human to human, both directly, indirectly through sewage into the environment, and then into the food that they consume or that the animals they raise consume. I was able to find one study directly comparing hospital effluent contents with farm effluent contents. And Dr. Noyes knows much more about this than I do, but if you look at in the top, let's see if my light works. I don't think it does. There it is. If you look at the top, this is actually work done by a group of researchers at Oxford who looked at effluent coming from an Oxford hospital compared to effluent coming from a farm, draining into the River Cam, and then finally river source water well separated from the two. And you can see that when they did their genomic study, and all they're measuring is antimicrobial resistance gene abundance normalized by bacteria presence as measured by 16S sequences, comparing that between hospitals and farms and river source. Obviously, hospitals are wonderful places for antimicrobial resistance genes to flourish as compared to farms, and certainly the river source water is very, very low. When they actually looked at mobile genetic elements, like of those resistance genes, how many were contained in the highly transmissible plasmids, transposons, you can see that the hospital species were considerably higher in terms of content of mobile genetic elements than were the species isolated from the farm or the river. But even there, that's farm related. And even in the farm, these mobile genetic elements were being selected for. So as you know, people give antimicrobials to animals for a number of reasons. Like humans, we treat individual infectious conditions used for as prophylactic measures at specific stages of growth. Metaphylaxis always interests me because actually we take the population of kids treated for what look like sore throats or ear infections, probably treating herds of kids with antibiotics, only some of which really have the disease we think we're treating for. And finally, growth promotion, which I understand remains banned in the EU. I'm not sure what's happening here in the US. I understand there are voluntary guidelines that are now being rescinded. But an interesting aside I will mention is that there is good evidence that antimicrobial use in infancy is actually a risk factor for obesity in later years, in addition to inflammatory bowel disease and a number of other conditions. But that's another story. So what is antimicrobial stewardship? I like this definition because actually it's species neutral, as you notice. It's just as good for you as it is for me. A steward engages in and designs and engages in processes which are intended to measure and optimize the appropriate use of antimicrobials by selecting the appropriate agent, dose, duration of therapy, and route of administration. So the objectives are obviously to achieve optimal clinical outcomes, as yours are to achieve optimal outcomes of as many healthy animals as you can, while minimizing toxicity and other adverse events, and minimizing the development of antimicrobial resistance. In my plain thinking way, basically using as little antibiotics as possible, although, as Dr. Noyes points out, there are many other factors that go into the emergence of antibiotic resistance. And finally, reducing excessive costs. But I can tell you from my standpoint of being a hospital steward, this is the one that cuts the least ice with physicians. It cuts most ice with administrators, but it's often the hardest to show. It actually takes more money to do the right thing often in terms of limiting antimicrobial use. So the team that I work in, and this is not in any particular order of importance, I want to emphasize, involves an infectious disease specialist like me, that may be a veterinarian in your case, an infection control and prevention specialist, the people that monitor the circulation of antibiotic resistance throughout the hospital, and certainly point out areas where we have fallen down and actually managed to transmit infection within our hospital, a pharmacist who is a drug specialist, a drug behavior specialist, who can assist us in making sure that we are only giving as much as we need, as often as we need it, a data analyst, IT support. This is the one thing I wish I had more of. The more that we work on the electronic medical record, the more data we generate, the more important it is to be able to reach into that data to see what we can do with it. A microbiologist. We are fortunate Children's have a PhD microbiologist. And finally, the unheralded members of the team, the virtual team. These are the stakeholders, basically the people that talk to me every day, point out cases I may have missed, ask questions that I should address. They're community pediatricians, or the hospitalists who take care of the kids in the hospital, and the intensivists who take care of the sickest. The tools we use are some of the tools that you would use. For example, local and national guidelines. For me, it's pneumonias, treatment of urinary tract infections, osteomyelitis. We use antibiograms intensively. We can tell you with pretty good assurance whether an E. coli encountered in the ED is going to be sensitive to this antibiotic or that antibiotic, which may vary very greatly from the susceptibility of organisms that we encounter on the floor. For the antibiotics that we can't easily substitute for which there is no alternative, there is pharmacy restriction. I have sometimes the ability to do chart reviews on everyone who receives antibiotics. When I talk about prospective audit and feedback, these are often going back into the charts and then parsing out the clinical details and then going back to the provider and discussing alternative ways of treating the child. Everyone, even if I don't talk to them in Children's, engages in what we call a 48 to 72-hour antibiotic timeout, which is when they circle back and look at culture results to determine whether they need to change their antibiotics or continue what they've used started empirically. I love this phrase, academic detailing. You know, for a long time, human medicine was plagued by detailing from pharmaceutical companies, which had its issues. Academic detailing is intended to be objective discussion between physicians, either maybe as groups at grand rounds or individuals one-on-one, about indications for antibiotics, side effects to look out for. And finally, hand hygiene and isolation precautions. As high-tech as we sometimes go, low-tech is king. If you're not washing your hands, no matter what fancy antibiotics you're using, you're working with one hand tied behind your back. Isolation precautions would refer to use of gowns or gloves or masks, N95 masks for TB, for example. The more I do this, the more I'm aware, though, that antimicrobial stewardship, when it comes to treating patients, fails unless great attention is paid to diagnostic stewardship. We have many antibiotics that we can use empirically, and we do so, but the important thing is not to use them empirically forever. We are using them only until we make an accurate diagnosis where we can use just the antibiotic with the spectrum of efficacy that we require. So this is really, for me, the basis of antimicrobial stewardship. And it's getting easier and easier. There were times when in a culture-based system, we were hamstrung by the fact we had to wait for the cultures to grow. Now we have DNA technology, and we can make quick and accurate diagnoses and avoid days of broad-spectrum empirical antibiotics. I point out vaccines because not only does good vaccine use prevent antibiotic use, obviously, and an example of that in my world is Haemophilus influenza type B as a cause of meningitis in infants. I came of age just as the wards full of kids with meningitis were going away. This allows us to tailor our differential diagnosis so we avoid using antibiotics to cover things that we don't have to worry about anymore. How do we measure success in the hospital? This is our Achilles heel. There aren't really good primary outcome metrics. We have a lot of secondary outcome metrics, which is basically days on antibiotics, reduced use of certain classes of antibiotics, reduction of home IV use, say substituting oral use for home IVs, because there are toxicities associated with delivery of IV antibiotics, for example, through line infections or clots, for example. But in terms of primary outcomes, perhaps the best we have in the hospital is a reduction of secondary bacterial infections that we know are directly related to antibiotic exposure, Clostridia difficile infection. How do you measure success in the veterinary ASP world? My understanding is, and Dr. Noy's talk touched upon this as well, is that there's a great deal of difficulty in relating change in environmental risk, resistant bacteria carriage by the animal, to clinical impact, illness in the humans. What effect will your change in antibiotic use or feed composition make to my patient in the end? Very, very difficult to make that connection. Because, as you know, colonization is not infection. Colonization can be transient. People be colonized for hours, minutes. I deal with short-term MRSA colonization, which may last for weeks, but we know from detailed research studies that colonization can be long-term, can last for years. And there's always a question of, is this a common source? Is the infected chicken and the infected human just exposed to the same common source in the environment elsewhere? A very insensitive metric, but one that you have to use necessarily, given the breadth of what you do, is gross weight of antibiotics used in animals. Unfortunately, it's not, reduction in use for growth promotion in the EU has been overshadowed by overall increase in antibiotic use. So making the direct connection between decreased use in antibiotics and health outcomes in humans has not been possible. Then there's the question of interventions. How quickly do we see an effect? Is the absence of an effect within an observation period of a week or a month really the absence of effect? Have we just not observed long enough? And I found an interesting article, for example, on the efforts made to decrease overall antibiotic use in German and Danish swine farms, where they did not see within the time frame of their study a change in the overall pork gut microbiome. But then it was pointed out that heavy metals such as copper and zinc were present in the feed, and that there are a lot of antimicrobial resistance genes that are actually repurposed, for instance, to work as efflux pumps to actually pump out excess copper and zinc, which I presume are not conducive to bacterial well-being. There are bright spots, and the Danish again lead in terms of interventions. Their decrease in macrolide use in Danish swine eventually translated to decreased rates of macrolide-resistant Campylobacter isolated from their animals. This has also been true in the human world. In the 80s, there was a growing problem of macrolide-resistant group A strep, identified in both Denmark and Japan. Concerted effort was made nationally to reduce macrolide use, and within several years, there were increasing rates of macrolide susceptibility noted. One Health, much is made of One Health. I find this interesting because these are 12 priority actions identified by a bunch of people who gathered to address healthcare-associated infections. They weren't talking about animal-related infections or overall planetary health. They were concerned only about humans. And interestingly, 5 of these precepts involve directly veterinarians, animals, and the industry that is involved in the production of animal foods. I want to point out the last one, which I think is important because this recognizes the reality. We need to find new economic models that reconcile public health interests with interest needs for profitability. So the agents of change that I see in my world and your world are pretty similar. I have consumers. They're patients. They're the parents of patients. I have many, many more parents now that are actually very reluctant to use antibiotics even when they're indicated. In your world, there are the consumers who are asking for antibiotic-free meats. There are the corporate consumers, perhaps like McDonald's, who are trying to source antibiotic-free beef. There are producers both at the local level as farmers and those who are contracting with the farmers like Tyson's, who are trying to contract for antibiotic-free chicken. Maybe a dirty word, but government plays a very important role here for a number of reasons. One is that's where policy is made. That's where legislation is passed and regulations promulgated. But I think perhaps the most important role of government may be the fact that they provide oversight, not in the sense of the oversight, punitive oversight or judicial oversight, but, for example, and I don't know what the equivalent is in your world, for me, the CDC occupies a very important role in terms of being a national clearinghouse for data that's important to me. CDC bankrolls studies, for example, national studies of resistance. That could not take place without deep pockets and a national focus. There are alternatives to antibiotics, as you know. I think you folks do a better job of this. Vaccines can be expanded. We're starting to see more use of bacteriophages directly or the active agent within bacteriophages, the organisms that prey on species-specific organisms, species-specific bacteria, phage lysins. There's more and more use of prebiotics, probiotics, and symbiotics. Prebiotics being the substances that promote beneficial bacterial growth, the probiotics being the beneficial bacteria, and symbiotics, the combination of the two. It is a rare patient that I see now that doesn't come in rightly or wrongly on probiotic supplements, and certainly when I use antibiotics, I make a point of using probiotics. Manipulation of feed components plays an important role. It was very interesting to me, for example, to know that E. coli 0157 carriage could be reduced by altering feed components in cattle. And finally, genetically bred resistance. This will be a tougher task, but there are already avian influenza resistant strains of chicken. Perhaps even more important, though, I have to say, getting back to the basics, sort of the global equivalent of good hand washing is a focus on infrastructure as a part of a water sanitation and hygiene initiative. We need to refine and rethink wastewater management, knowing that both bacteria, their genes, and antimicrobial agents are essentially just being recirculated over and over again using the current methods that we use. We know that anaerobic digestion and Lyme stabilization are more effective than UV and chlorine treatment in reducing bacterial content in human waste streams. And there can be some low-tech solutions, basically placing rain covers over manure piles to reduce a runoff from the piles into local water sources. There will need to be investigation to identify hotspots of increased antimicrobial resistance gene burden, so you can focus specifically on areas, for example, in pig farming, the early finishing pigs, which have a higher load of ESBL Enterobacteriaceae colonization, or in slaughterhouses. So the lessons I've learned from ASP in the hospital, the core challenge is to change a prescribing culture. Antibiotics are an anxiolytic, as you know. You feel better when your patient is being treated. It's also hard not to treat an animal or human when you have been told by people older and more experienced than you that this is how one does it. Stewards are best coaches and not police. They are collaborators and mentors. I want to emphasize this is a team effort. Every member of the team contributes something uniquely to the effort. There is no one person who has a monopoly on the truth. And perhaps most importantly, every success needs to be shared because each success will feed the next. Communication is key both within the hospital and between hospitals. We spend a lot of time querying each other about best practices.